Okay, so the last topic I want to cover in this Genome Browsers session is data export with BioMart. So what I'm going to talk about is what BioMart is, what you can use it for, and the basic four-step principle. So BioMart is a data export tool. There is absolutely no programming required whatsoever, and you can generate quick tables with it, which you can manipulate in tools like Excel. Just be aware before you open any spreadsheet with gene names in it in Excel that it may try and change some of your gene names such as OCT4 or SEPT9 or DEC1 into dates. So look out for that. And BioMart's basically a web interface that allows you to export tables of custom tables of ensemble data. So why would you use BioMart? Well, if you're working with a lot of genes and you want to get information about a lot of genes, using the normal browser is kind of like doing your shopping by going to lots of individual shops. So going to a bakery and going to a butcher and going to a greengrocer. Whereas using BioMart is like going to a massive supermarket. You can just get everything in one go. And you'll find BioMart at the top blue bar of all the pages in Ensemble. It's available for Ensemble plants, most of Ensemble fungi, Ensemble metazoa, and most of Ensemble protists. And what can you do with BioMart? Well, if you start by defining a set of genes or variants or regulatory features by combinations of parameters, so a region or a list of IDs or a function. And for that set, you can export data, IDs, sequences, homologs, in a really nice table. And it consists of 4 steps: dataset, filters, attributes, and results. So dataset is defining which database you actually want to search. So this is the genes, variation, regulation. There's also another database in there called mouse strains, and that's looking for genes between different mouse strains. And you define the species. So you might pick genes and then human, for example. Your next step is filters. By defining your database, what you've then said is, right, I'm now looking at all the genes in human. You want to filter that down to the ones that you might be interested in, which could be by putting in a genomic region or a selection of genomic regions. It could be a list of identifiers. It could be a Go term, and you want to get all the genes which are associated with that Go term. It could be phenotypes. And if you use multiple features, they use AND logic. Then you get attributes, and this is basically defining the data you want to export: IDs, features, sequences, orthologs and paralogs, coordinates, all these kinds of things. This is actually what you want to print in your table. And finally, you get your results, and you can either get a table, which can be HTML, or it can be tab or comma separated, or as Excel, taking the warning about gene names, or you can get sequence, which comes as FASTA. If you use R at all, there is an R interface called BioMart that you can use, and I've just included this to point you to the documentation. If you get the downloaded copy of these slides, you can go to the links here to get the documentation in order to use it, but I won't go into detail at all on actually working with it. The principle of what you're trying to do is the same, but it's just you do it on the keyboard rather than with a clicky mouse. If you're working with BioMart, then we do have a paper with more information about it. So what we're going to do, the example query we're going to do with BioMart is we're going to look at a list of 6 genes. And with those 6 genes, we're going to find out their NCBI gene IDs, their function as Go terms, and their cDNA sequences. So if we take our model here, with our dataset, filters, attributes, and results, a really useful thing to do when you're starting out a BioMart query is just to go through the question and go through the task you want to do and work out actually what is my dataset, what is my filters, and put these into their categories before you get started. So in this case, our dataset is genes, and I haven't put it in the question, but it is in fact human genes. Our filters are things that you already know. And so in this case, it's our list of gene IDs. The attributes are the things that we actually want to find out, that we want to print in our table. And one of the things we want to print in our table is probably our initial input, but as well as that, we want our Go terms, our NTBI IDs, and our cDNA sequences. And the results will just be our tables. After we've gone through the demo video, then there's a task that you can do as well.